All right. Hello, everyone, and welcome to our final session of the day. It is my pleasure to introduce our second keynote speaker, Dr. Neil Fleshner, co-founder and chief medical officer of Point Biopharma. Dr. Fleshner is a scientist, clinician, and entrepreneur. He graduated in 1988 with his doctorate in medicine from the University of Toronto, where he also completed a residency in urology. He then spent 3 years at Memorial Sloan Kettering Cancer Center, where he also completed a master's in public health at Columbia University. He has been actively practicing urologic oncology for 25 years and has published over 500 scientific manuscripts. He has also secured over $50 million in peer-reviewed funding. Dr. Fleshner holds the Love Chair in Prostate Cancer Research at the Princess Margaret Cancer Center, as well as being immediate past chair of urology at the University of Toronto, where he is also a full professor. In 2020, Dr. Fleshner co-founded Point Biopharma, which actively pursues theranostic medicine and went public on NASDAQ in 2021. And with that, I pass it off to Dr. Fleshner.
Well, thank you, Nick, for that nice introduction. And I want to just thank the organizers for having me here today. It's quite funny here, my disclosures, for me to give this talk because, as you can imagine, I've spent most of my career giving talks at medical conferences, more recently at biotech conferences, but it's great to have more of a diverse audience. Having said that, some of my slides are a little bit medical, but I hopefully will be able to explain them in fairly plain English for many of you. So to me, this is a very interesting sort of slide that tells us why precision medicine and why medicine itself is getting so complex. And this was a study done about 10 years ago where they plotted the doubling time of medical knowledge. And basically what you could see is in 1950, what took 50 years to double all of the medical knowledge in the world prior to that, it was 7 years by 1980, 3.5 years by 2010, and is now projected to be 0.2 of a year, which is 73 days, meaning virtually every 2.5 months, we learn more about medicine than we've ever learned in the history of humankind. And as you can imagine, that creates this amazing body of knowledge for us to draw upon for new insights to better diagnose, treat, prevent diseases. Of course, it also causes a huge perpetual gap of knowledge about what we think. Again, by 2 months, you're almost outdated in the field of medicine. When we talk about precision medicine, I know you've had some other talks about it in this meeting, is really, and there are many different ones, but the one I like is an approach to identify using environmental, lifestyle, and biological approaches, a treatment strategy that will lead to better disease outcomes. And that's from the NCI Dictionary of Cancer Terms. When we talk about a biologic approach, there are many platforms, and this continues to expand, right? They can be phenotypic, meaning the ultimate picture of something. They could be protein-based, right? They could come from a blood test, a saliva test, an imaging test. It could be omically driven, and I'll give you an example of that with a drug called olaparib, which is used to treat advanced prostate cancer, or it could even be immunologically based, and I'll also give you a little example of that and why this has been so amazing, but also why it opens up some controversies as well. For those of you not familiar with omics, you've heard, I'm sure, the term genomics or proteomics, and really what it is it's a high throughput platform that simultaneously can measure the totality of any molecular process. You could simultaneously, as you can see on the right, this is a gene chip, so you can measure simultaneously thousands of genes and what they're doing in a cell at any given moment, and by turning it to the upregulated genes is the green, the downregulated genes are the red. You could not only learn what genes are players, but you could also start fingerprinting them or thumbprinting them by pattern recognition to get an idea of how these work. And I think when we look at the personalized medicine revolution and why it's spurred on so much in the last 15 years, it's because of computing and high throughput omics, which gives us the ability to really have a powerful way to measure processes that we never could before. It's an interesting example that Nixon declared war on cancer in the 1970s, but not much was really accomplished in the first 40 years. And the reason for that is the analogy I give is we were fighting a nuclear foe with effectively bows and arrows. This puts us on equal footing to study the complexity of human medicine and the biologic processes therein, to really make us accelerate discovery. Having said all that, precision medicine is not new. It's been known for over 200 years that you can't just give the blood from any stranger to an additional stranger. And to some degree, that was probably what's considered the first, if you will, use of a precision medicine, this recognition that certain bloods would clot if you put the wrong blood types together. But particularly in cancer medicine, which is what I study, it has taken significant traction, again, over the last 15 years in particular, due to high throughput omics and our ability to measure and the bioinformatics that it leads to. And the hope, and I think it's bearing out quite well, is that this is leading to more efficacious and less toxic therapies. Now, it's interesting. I mean, what is the traditional model of pharmaceuticals or medicine? One was the single arm study, right? Where you would just take a group of patients with a trait, say prostate cancer, give them a drug and look for a response. And if you saw a response in a reasonable proportion of that, you would say that this is an effective drug in that space. Apologies for that. But what if in reality, say 10% of patients are super sensitive to the drug and 90% are not? You would dilute or drown out the recognized benefit in those 10 of 100 patients who otherwise could have had an amazing response. It just wouldn't be recognized. The other models sometimes are comparative studies. You take drug A and you compare it to drug B, and if overall A is better than B, then A is now adopted as a standard of care across the world. And this was how cancer therapy has been going the last sort of 30 years. And the problem with it is we rarely make leaps and bounds of advances. They're usually small steps. So when they're small steps, it leads to interesting sort of a math exercise. If you gave drug A and it cured 100% of people compared to drug B, which cured none, you wouldn't need all this complex mathematics and study. But the reality is it's usually just a little bit better. So let's say treatment A is 5% better than treatment B. You then have to ask yourselves, is it that every patient derives a 5% benefit? Is it maybe that half of the patients get a 10% benefit and half none at all? And even in a negative basis, what if a third of patients derive a bigger benefit, and a third have no benefit, and a third, however, can be harmed from the treatment? So you can see all these scenarios can play out, and really personalized medicine offers us the promise of fixing this problem, really trying to identify who benefits, who doesn't benefit, who may be harmed, and try to identify and pluck out the responders and non-responders as soon as possible to afford better outcomes. I'm going to use an example for you, and I'm going to try not let it get too technical, but this is a class of drugs called PARP inhibitors. This is something from my neck of the woods in prostate cancer. And these are a class of drugs that inhibit DNA repair. And it's interesting that if you give 1,000 men with advanced prostate cancer these drugs, you will barely see a response. But it was subsequently learned that there are subsets of men with advanced prostate cancer that have defects in their DNA repair who can respond. And really it reflects a small proportion. It's maybe about 20% of patients, of course, not all of them respond. They have alterations in their DNA repair. So their ability to repair damage of their DNA is impaired in the cancer cell itself, which, as I'm sure you all know, continuously gets mutated. So it opens up the possibility of something called synthetic lethality. It's just a term, but I'll try and go through it with you. Essentially, to repair DNA, you need to have good PARP function and good what's called BRCA. You've probably heard of the BRCA gene. BRCA function is very important in repairing DNA. In a normal cell that does not have a BRCA mutation, if DNA is damaged, it will repair the problem either with or without the presence of a PARP inhibitor, because you need to wipe out both PARP function and BRCA function to kill the cell. If, on the other hand, you have a normal cell with a BRCA mutation carrier, a non-cancer cell, it only has one allele, right? We all have two forms of every gene in our body, one given by mom, one given by dad, or it could be a new mutation, which commonly happens in cancer. But if only one of the pair is damaged, which would typically be somebody who was born, for example, with a BRCA mutation, then if you damage that cell's DNA again and give them a PARP inhibitor, once again, the cell will survive because BRCA function is preserved, because you only need one copy of that gene to have enough function. It's only when both alleles are lost, which happens in certain cancers, and you inhibit the PARP, then you get cell death. So this leads to this sort of scenario where if you give this drug, it doesn't matter the name, to patients who have prostate cancer, who have the biomarker, meaning they have that DNA repair defect, they do extremely well. Whereas those who don't, effectively, that's a placebo effect. They have no beneficial effect. And this translates both into delaying progression of the disease on x-rays and in improving overall survival. And this led to the approval of this particular drug in the subset of patients who have DNA repair defects. So again, otherwise, without these tools or these biomarkers, this drug never would have been a candidate for men with prostate cancer. And this dovetails to radiopharmaceuticals, which is the topic of the company that I founded, Point Biopharma, where radiopharmaceuticals have been around for a long time. Since the 1940s, we've used radioactive iodine to treat thyroid cancer. But in prostate cancer, the radionuclear drugs were of limited use. You can see them there. They were not particularly effective and were very difficult to manufacture. They were hard to make and administer. And the pharmaceutical companies did not embrace it, mostly because of manufacturing and logistics. So imagine, to use a radioactive drug, it decays. That's the nature of radioactive physics. So some drugs may have a half-life of a day, which means when you make it, you cannot, if you will, have a product that's manufacturable at a central location and distribution friendly across the world or even across a small area because of the basic physics of it. So this is what really hampered this industry until about the last 5 to 7 years. And that's how we got involved as well. And this was our key entry into the field, a molecule called prostate-specific membrane antigen. And this is obviously important in prostate cancer. What's important about it is it's a molecule that sits on the surface of the prostate cancer cell. And it basically sits like a pocket, if you will, where molecules can bind or touch it and bind with it outside of the cell. There are a variety of different PSMA ligands or if you will, molecules that actually bind to that. But some very clever scientists, mostly in Europe over the last decade, started to develop these molecules. And really and I'll go through it with you again, what's key is they bind the PSMA molecule. And then they often have this pocket where you could then put a radiometal in there. And why that's important is the type of radiometal can then tell you what type of where your cancer is and maybe even treat the cancer. So I'll show you how it works. Gallium is a radiometal. So if you put gallium into this little pocket here and inject this into the body, this molecule will travel around all the molecules and will stick to the prostate cancer cells and emit what's called a positron, which is the molecular or the the physical particle that gallium emits. And that's the basis for a PET scan called positron emission tomography. So you could then take pictures of this, and this poor unfortunate man, you can see each one of these dots, not the big ones because that's the kidneys, but all these dots are deposits of prostate cancer in that man's skeleton. Okay, so this has revolutionized the management of prostate cancer because it's much more sensitive than traditional imaging, like just regular CAT scans or other scans that are done. And this is another example of a man. This is with a lymph node affected by prostate cancer. You can see it light up on this particular imaging. Now, once you can image it, the question is, can you now really apply a personalized medicine approach and say, well, can we also treat it? And that's where now we get the concept of a therapeutic isotope. So that this would be a drug that now is not only used to image the tumor, but actually used to treat it as a bomb, if you will, smart bomb. So you could attach an isotope, the one that's most commonly used, lutetium-177, that can actually deliver radiation to the cell and kill it. So you have this nice pair of and that's why it's called theranos. You have the pair of the imaging, and then you have the therapeutic together. And I use this analogy a lot, this keychain analogy. When you think about a keychain, you have the part that fixes into the lock. So this would be the motif that would bind to the PSMA. You have a little linker, and then you have, in this case, it could be gallium because this is your imaging marker. But now when you want to do therapy, you just swap out that gallium for a therapeutic isotope like lutetium, and now you have effectively liquid radiation. And there's much less side effects than traditional radiation because it only will bind to the cancer cells with very minimal off-targeting of normal tissue. And then you can start to see this. Again, this I just showed you, this is the PET scan, but look what happens after it's treated with our product. Most of the disease actually disappears. So this is a man who's riddled with cancer. And after 3 or 4 treatments with our drug, you can see the cancer is gone. These dark spots are the kidneys and how the drug is taken out of the body. So that's why that doesn't change. But this is like a miraculous type of response to our therapy. So the PSMA was largely discovered in Europe over the past decade. And what was happening is doctors were making it in their hospitals. We call it homebrew, in the basement, and they were troubled by the same thing I just mentioned, how to make it well, how to make it according to pharmaceutical standards, and how to overcome the half-life. So these drugs were made in the basement, very little quality control, not even basic toxicity data. And as I mentioned to you, the half-life was 6 hours. And one of the things that we did at our company is we've built a patented ring fence around that, extending the half-life now to 1 week. And once you have that, you could have a just-in-time manufacturing plant that can globally even distribute these types of drugs around the world. They are expensive, however. The isotope alone is about $4,000 to $5,000 per dose. So commercial interests started to overcome some of the challenges that was found in the basement of hospitals in Europe. AAA or Novartis came in, and ours came in. We started to do proper trials that were more in a traditional pharma model. And as I mentioned, we were able to alter the half-life, allowing for commercialization. And then trials were done. This is a chemotherapy drug that we know works in prostate cancer. And this molecule, this lutetium PSMA molecule, this is Novartis's compound, was shown to be superior than a chemotherapy that we knew worked in this setting. And similarly, now overall survival, so life extension as well, now realized using this type of product. So I think it's an interesting story in that these were sort of discovered or developed, if you will, in academic labs, but really required big pharma to take it to the next level. So this is what sort of the PSMA space, and this is version 1.0 of what's an opening space. And now you're starting to see new clinical trials, new ligands, and I'll show you a little bit about that. So our product, our 0.202, is now, instead of going after chemotherapy, we're trying to bring this cancer treatment up earlier before patients get chemotherapy. And the whole idea here is we actually de-escalate their therapy. We take them off the drugs that they're starting to fail. We give them 4 or 6 injections over a year, and they seem to be remaining quite well. So this is actually a big step forward in men's health, with the idea of ratcheting up toxicity as patients get sicker and not use the less toxic therapies later. But there are some interesting questions that arise when one embarks on any new treatment. And I'll give you the example here is we rarely know the long-term side effects, right? It's come out a lot around the COVID vaccine. and the first generation ligands that I just showed you can have some off-target activity of the salivary glands that make our saliva, the lacrimal glands that make our tear, and the kidneys. And the reality is most radiation-based damage doesn't affect somebody clinically for 5 to 10 years. You would never notice this abnormality because patients don't live that long. As much as we're trying to help them, we're not curing them. And many of them, if we use the same drugs earlier, we will start to see, dialysis requirements and other problems arise as an attempt to make patients better. So what we are doing as a company are strongly investing in the next generation PSMA molecule. We've already have our lead, which is called this PSMA 62, which actually has less kidney uptake and 3 times the binding at the prostate cancer cell. So that allows you to reduce the radiation dose, if you will, by 2/3, and you would have effectively the same treatment. So these are the ones that we feel can go earlier in the disease. And certainly as a surgeon, my vision to take patients after surgery or radiation are high risk for micrometastatic disease or disease that needs to be mopped up, and use these drugs to try and do that, very similar to thyroid cancer paradigm. And then we also have other really cool technology. We're studying this now preclinically in our company to again, how to minimize the fact that there can be off-targeting activity of our compounds. And this is called what's called a prodrug construct. So I don't want to get too technical, but basically in the cancer microenvironment, but not in normal tissue, you have a second molecule expressed called FAP. And what happens is we could build a molecule that basically hinders the radiopharmaceutical drug from being bound to the tissue, but only will do so where the FAP is present. So in the cancer tissue, it will be deployed, whereas in the kidneys or salivary glands, it won't be deployed. And again, trying to minimize off-target activity. And I think this in the next decade or so will be a major player in a variety of different cancers. It's a pro-drug idea. Exciting times, but there are controversies. And I do want to touch on them that we're going to have to figure out as a society and also as a medical community. And a lot of it has to do to some degree with reimbursement and making things better. So the first is this concept of the biomarkers. Are the biomarkers really that good? So another miraculous group of precision molecules have been these immune checkpoint inhibitors that are used to treat cancer, particularly, I mentioned one earlier, pembrolizumab. There are others. These have had an amazing impact. When I was a young oncology doctor, if you had a metastatic melanoma, it was 98% fatal. It's now, we're curing about 60% of patients because of these drugs. And what they do basically is we've known that cancer cells evade the immune system. They do that by expressing on their surface a molecule called PDL1. It's not that important, but what PDL1 does is dampen down and inactivates the T cell, which is the immune cell, shuts it down. Not surprisingly, the cancer cell would in a Darwinian pressure type of environment would express this in order to survive. But what's interesting is, I don't want to make this too complicated, but here's one of these drugs. And the hope was that if we could take the tissue and interrogate it and see, because you can do some research on the tissue, you can stain it and see, does it express this PDL1? And the answer is yes. If the more it expresses it, the higher the response rate are. But the problem is it's not hugely discriminatory. So if you express it high, it's 26% response rate, but if you express it low, it's 8%. So it raises some interesting questions. It's not like it's that great in these guys. And what about the 1 of 12 who would respond, in whom you would have thought historically the tests would not work? So some respond well, but how to handle these biomarker negative patients continues to be a problem, particularly for payers who don't want to spend that particular drug is about $12,000 a month. It's $144,000 a year of therapy. Are you going to want to spend that on a patient who may not respond? But how do you handle if it's a 5 or 10% response? It's not zero. And this just shows you, here's PD-L1 testing in some diseases, because it's used in many cancers. It's not predictive. Others, it's predictive. Others, it's predictive and approved. In other words, like a Siamese twin, it's linked to get approval for the drug. So again, the biomarkers are not perfect. And here's another example. I showed you this curve earlier. This is the curve from the PSMA therapy. These patients are highly selected. They get a PSMA scan, it lights up. They should respond. But look at this. The curve drops down in the first 3 months. These are non-responders. About 20% of patients exposed to these drugs don't respond. Why is that? So obviously the biomarkers aren't perfect, and usually translational medicine will help us sort that out to figure out who to best select. But again, the promise of precision medicine is there, but it's never perfect. A little diversion to another, if you will, controversy that will come up is, what about all these biologic specimens that we've been talking about? You can't do any omics. You need a biologic specimen in order to do it. And I'm sure many of you are familiar with the story of Henrietta Lacks. If you're not, this was a Black woman in the Baltimore area in the 1950s. She developed vaginal bleeding, was diagnosed with cervix cancer. She was treated at Hopkins, which, by the way, in those days, very few hospitals would actually treat underprivileged people, and they did. A surgeon took a biopsy of her tissue, and unlike hundreds of others' attempts, her cells grew in a test tube and a Petri dish way better than anybody else's. And that cell line has now been propagated and is still used in research. You could see this 70 years later. And in fact, they actually called it the HeLa cell line, which is from the two first letters of her name. The sad part of it is Hopkins and the world benefited tremendously from her gift, if you will, want to call it that, to science. But she never did, and her family never did. And there's a whole story in a book about it, how many of them were left poor and destitute. And to some degree, she is also a big contributor to cancer research in her own way. So I think this is an interesting question, particularly as patients get more and more tech savvy. Everyone's more and more attuned to the power of data and big data. And of course, that's what this ultimately is, right? We're converting biologic samples or imaging-based outputs into big data. And I'll just make a little plug, if you will. This is a slide given to me by Mariella Gross at the University of Pittsburgh. And she has a vision to really engage donors of biologic samples using a non-fungible token to actually, as a way to get them to have some ownership over the biologic specimen that they have donated for research, so they can see the fate of the sample, and they can see the what's come of it and what discoveries even have been made using it, with the idea right now of mostly information purposes. But one can imagine if there could ultimately be even of some sort of financial kickback. And the beauty of the NFT is you can use it to descramble the identity of the sample and then rescramble it back for the end user, in this case, the patient who would donate the sample. And her vision is a patient would just get a bio wallet, if you will, like basically an app on their phone, and they can really see how it goes. Really nifty idea at a very early stage, if you will. The last thing I want to just touch on is cost, because this is the other issue. Is of course it usually takes somewhere between 5 and 12 years to bring a drug to market. Usually people typically say $1 billion. I think it's probably more today. And I did this very cursory analysis. These are the drugs I use to treat my patients with advanced prostate cancer. These are US prices, but we give these drugs, which are drugs to basically chemically castrate men. These were they're $36,000 a year. When I started off in urology, this was considered heresy. This was eating up a huge part of it. In Canada, our provincial budget for systemic therapy for cancer patients, it seems really cheap now. The next level, hormone drugs, typical duration of therapy, 30 months, is $432,000. Radium-223, another $170,000. Chemotherapy is cheap. Our drugs that we just talked about, another $250,000. Why not? And let's throw in some olaparib for some of the patients. So you can start to see what happens is that for a man to check out of planet Earth with advanced prostate cancer, and he's not being cured, he's not going to contribute much economically. These are usually men who are older. But you're looking at maybe upwards of $750,000. And somebody has to pay for this. And ultimately, it's going to be the taxpayer, or as a society, we're going to have to decide how to get the best bang for that dollar and how best to use precision medicine. And really, these promises of returns is what keeps the capital markets alive in biotech. And you have to ask yourself, if this is fiddled with it anyway, and there has been some, as you know, some moves to cut drug pricing through the Biden Congress just a few months ago, how will this affect discovery and the whole industry around discovery and startups? If the long ball isn't there for some sort of promise of massive return, particularly given the fact that many of these are speculative and high risk investments. Here's some others. Hepatitis therapy to cure Hep C is $100,000. CAR-T, there's an article in New England Journal of Medicine just this past week, about $1 million now upwards a patient. And this hemophilia gene therapy, $3.5 million. So again, I used cancer as my first example, but here's some nice examples even from the non-cancer world. CAR-T is a bit of a cancer drug, but it just goes to show you how this whole field is evolving, and it's staggering. It's easy with a rare disease, but staggering when you get a common disease. So I'm just going to wrap up. I think I'm right on time. Just tell you, first of all, that precision medicine clearly is here to stay for good. And as I showed you, I think knowledge is going to keep moving fast. We will have more targets identified. They will make the world a better place. We'll have less toxic treatments. We'll have more efficacious treatments. But there are very important societal considerations here. As I mentioned, are the biomarkers good enough to discriminate responders and non-responders? And if that's the case, does everybody get it? In which case, it sort of starts to defeat the process. Or what are fundable thresholds from a third-party point of view that say we will cover this? Despite all that, we have to still ask ourselves, who is going to pay for it? Certainly, the dollars involved are huge. The sums are huge. Even wealthy people, it's a huge amount of money. So this is beyond a class thing. This has to be a societal thing. And as I alluded to, if we decide as a society we're not going to pay it, to what degree will capital markets then squelch the pace of discovery, or at least maybe not so much the fundamental discovery, but the translation from fundamental discovery to the market. And then lastly, I do think as an equity issue, we have to start asking ourselves who owns the samples that are biologically associated with discovery, and how should we handle those individuals who in their own way have contributed? Is that all an altruistic model, or particularly in a system where patients are paying out of pocket, can there be some sort of restitution? So I'll leave it there. Again, thanks. I hope I was not too science and geeky for you, but I also wanted to we'll go engage in a little discussion. So thank you.
All right, great. Thank you, Dr. Fleshner. We'll start the Q&A portion now. So if you have any questions, just go ahead and drop them in the Q&A. We have a question from Dr. Addy, who is another urologist. He is wondering, is there any hereditary familial significance of BRCA mutations between male prostate cancer risk and women's breast cancer risk?
Huge. It's huge. So historically, the whole field of medical genetics, if a woman was diagnosed with breast cancer, was to only test the daughters. And nobody ever paid attention to the son. So we've just actually published a manuscript. If you're a BRCA2 carrier in particular, which is extremely, which the breast cancers associated with BRCA2 carriers are actually less aggressive than BRCA1. It's the opposite in prostate cancer. We're now estimating that a BRCA2 carrier has a 40% lifetime risk of prostate cancer death. It's huge.
Great, thank you. So I actually wanted to ask you something that we discussed before we hopped on this call. You mentioned that in Canada, there's a 93% consent rate for tumor tissue banks. And it just kind of brought to light something I think would be useful for everybody here is just where do you think we can learn from other countries like Canada as us in the US are working towards implementing these new precision medicine solutions?
Yeah, I mean, I think to some degree, the US leads the way. And I could tell you now, I'm putting on my industry hat, our company's all focused on the US market. I think we spend 90% of our strategizing on the US market and 10% on the rest of the world, because that's where the big bang for the buck is. You've all read the news, how drugs are way more expensive in this country than anywhere else. And some would argue one of the functions of America and the NIH is, and this is what fuels, it's almost like a tax we all pay to fuel global entrepreneurship and innovation. But that goes only so far, right? When patients can't get access to treatments. So, we believe in Canada, and I personally believe it, that healthcare is a right, not a privilege. I know some Americans feel differently. And we as a country have taken the view that everybody is entitled to the best treatments out there. And it is wreaking havoc on our country to some degree, but we still spend way less than you do in the States from a GDP proportional point of view. But it is eating up our budgets in an aging population. So I think from a precision medicine point of view, I think the US has been much faster, frankly, to embrace it. We tend to be much more conservative, mostly because the payers, those who grant the purse, to open the purse to pay for the drugs, tend to have a higher threshold to say yes than you would in the States. So there are many drugs that are approved in Canada, in the US, and even frankly in Canada, but still not reimbursed by our healthcare systems because they don't bring what's perceived to be enough value to the patient and or the system. So I think it's this value-based idea of treatment, and you wonder if by doing that you're going to set a higher bar for therapy. The problem is, particularly in cancer therapeutics, when you disturb a cancer pathway or a cancer mechanism of growth, because it exerts a Darwinian pressure, and a new subset of cells will come that has now resistant to that treatment you gave them. So it's constantly this cat and mouse game, and because of that, we tend to get, as I mentioned, these baby step marginal benefits that are 4 months, 5 months, but then you start stringing 8 or 10 drugs together, and hey, now you're living an extra 3-4 years.
Yeah, that's very true. So I have another question from Dr. Addy, and I apologize to the non-doctors in the audience. It's a little bit technical, but he's wondering, would it be life-saving to have non-radiating MRI scans coupled with Galleri blood tests for multiple tumor markers? And how often should they be given, from when, and in whom?
Right. So again, this is, referencing some of the more novel ways of diagnosing prostate cancer. PSA tests he's referring to. MRI is now the newer kid on the block, which has been demonstrated to be better than our traditional ways of diagnosing prostate cancer. And I think we have to let the data bear it out, right? I mean, I don't know that we know right now what the right timing interval is for MR, what the right thresholds are. We were tending to use a PSA threshold. I can tell you getting back to the first question, my BRCA carriers, I do an annual MRI, not based on any data, just based on pragmatism. So we are, these are all great questions for investigation and CV building.
Wonderful. And I know we have some entrepreneurs in the audience. So I just, seeing as you're an entrepreneur in an incredibly capital intensive field, I'm just wondering if you could kind of give the entrepreneurs in the audience a little bit of advice as they're going through and, trying to develop their own solutions that may or may not be very capital intensive.
Yeah, it's a great question. A lot of people, I mean, how did I back into this company was quite interesting. I mean, it was a classic example of intellectual curiosity, a little serendipity never hurts, and having the right brains around you at the time. So I'll tell you how it all happened. I've always prided myself on being ahead of the game in my practice. And I was reading about some of these crazy cures. I showed you some of the scans, cures, responses, they're not cures, of patients in Germany. And I'm fortunate enough to have quite a carriage trade practice. And I had a few patients who were at the end of their rope. They had no other therapies, no other hope. I said, look, I showed them this. I said, if you want, I will take you to Germany. I'll be your personal physician. And I escorted 2 men. 1 week, it was in January, about 6 years ago. We went to Berlin and we treated them. And I was tending to them and their care, but in the back end, I was talking to all the folks at the Institute, learning about what they were doing. And I came home and I said, there's something real to this. And I started getting the balls rolling to open a center at my hospital, actually, but started to get a lot of gaps. But as part of that, I started interrogating what the local sort of landscape was. And it turned out that even though Canadian patients weren't benefiting from these molecules, one of the world leaders in manufacturing and distribution of the molecules was about an hour, 45 minutes down the road from me. Called up the CEO. We went and had dinner together one night, and the company was born. So, and we riffed it. Our third co-founder was a finance guy, but basically, if it wasn't for my curiosity to go look, finding this individual, then also having roadblocks in my own hospital, because I could have quit 50 times, it never would have happened. And so that was the origin of the story. Then, so we got together, created a business plan, little friends and family, Series A, and then we actually went public, in our case, through a SPAC about 2 years ago. So that's the story. It was quite an interesting one, but it's a classic. What are those stories, right? That if I didn't run into Joe McCann, who's our current CEO, who was the CEO of that non-for-profit company down the road, it never would have happened. I mean, there's so many stories like that.
And that's what they say. Serendipity is huge in entrepreneurship. So we have another question from Caroline. She mentioned that she's read about a shortage of geneticists across the US, and she's wondering if you could comment about how we can bring preventative precision medicine, such as genome sequencing, into the clinical setting without enough geneticists to interpret the results.
That's a great question. So thank you for it, Caroline. So I think there's a simple answer here. First of all, I'm pretty sure in 10 years, like now you would get a panel of 60 genes. I'm sure in 10 years, we're all going to have a copy of our genomes in our iPhones, right? Because the prices are dropping like crazy. And I think the short answer to your question is, and we've adopted this in Canada where we have even less geneticists, is a different type of model, which is the traditional genetics model was go talk to your geneticist, talk about the pros and cons of testing, and then decide whether to test and then go back if positive. The current model we're embracing is let's have the physicians do the first part. And then let's hand it off to the geneticist only if there's an abnormal result, an actionable result. So by this way, you leverage the physicians as the first line and try to preserve the genetics people. Because if you think about what they're doing, genetic anomaly is usually found, at least in my disease I study, is 12%. So which means 88% of their initial interaction is a waste. So I think this is the model that we should be embracing.
Great, thank you. So I wanted to say thank you to Dr. Fleshner, and thank you to the rest of our presenters and panelists. We also wanted to say thank you to all of the audience, and hope you've enjoyed learning more about precision healthcare. We've learned a lot about exciting new innovations, such as bringing design thinking to medicine, gene editing, holistic personalized data that's owned by the patients. And we've also touched on some next-gen technologies, such as AI, blockchain, and NFTs, and how they may or may not fit within healthcare. We've discussed some current challenges, such as the financial hurdles associated with switching value-based care and data privacy and transparency concerns. But we've also talked about some ideas to overcome these challenges, on top of some great new opportunities like reducing the trial and error in current treatment pathways. There's a lot of work to be done in precision healthcare, but there is even more to look forward to. And I'm excited for all of us to work together on realizing the potential of precision healthcare. Thank you again, and have a great weekend. And with that, I will close our 2023 Vitals Conference.